Fine. Yeah. So, hi everyone. So, first of all, I'd like to thanks all of you for registering yourself for this particular session, in which we'll be talking about one of the most important aspect of biological sciences, that is NGS, which is next generation sequencing applications in the field of DNA, RNA, and the genomics. And to cover this particular topic, we have with us Dr. Kunwar Digvijay sir, who will be giving his brief introduction right now. And after that, we'll be starting with the session. So before starting with the session, let me first just give you a little bit brief idea about us, that is who we are and why are we trying to conduct these sessions, what is our motive behind that, so that you will have a little bit good idea behind before registering these sessions for coming future. Okay, sir, can you please confirm my screen is visible or not?
Yeah.
Great. Fine. So, first of all, I'd like to welcome all of you to our official event, which is official event to the particular session. And we conduct these sessions twice a month. And this is the first event of this particular month. So, we basically belong to Techmedbuddy. So, Techmedbuddy is basically our platform whose motive is that we believe in the philosophy that change is the only constant and we should keep on changing, because you can't prevent yourself from getting changed, and your behavior will keep on changing. And this is what academia is also going to teach you. You will keep on changing with the time, and you should also. And Techmedbuddy is basically a platform which is trying to bridge the gap between academia, research, and the industry. So this is basically our team and the motive with which we are trying to take this initiative and move further. And these are the three wonderful people of our team who worked really hard day and night to bring this session before you, because of whom you registered to this particular event. So in today's event, we have with us. I'm really extremely sorry. By mistake, I put the wrong name. I'm really sorry. So
It's fine.
I'm really sorry, sir. So
No, that's fine, Alok.
yeah. So in today's event, we have with us Dr. Kunwar Digvijay sir, who is a senior postdoc researcher at University of Illinois in the field of clinical aspects of NGS applications. And this is me, Alok, who will be covering few aspects of the downstream processing of this particular sequencing data and all everything, because data generation is one thing and data processing and getting the output from that is another aspect. So I will be giving you a brief idea about what do we do being a computational scientist after getting the data from the wet lab. So today you have a team of wet lab expert and a dry lab. So I'm not an expert definitely in this particular field, but yeah, I have a little bit good knowledge in this particular field. So I'll be giving you the idea about the computational aspect of the data after we get from the labs. So the topic of our today's talk is NGS based application of DNA RNA in the clinical sciences. Along with that, we will be giving you an explanation of differential expression analysis and few other aspects. Yeah, and this is about us. So what we are trying to do, so we of course provide free of cost career counseling services. So if you are a bio oriented background student, or you belong to the field of data sciences, machine learning, or few other fields, feel free to reach us anytime if you want to make a transition to bioinformatics or data science approach to biological sciences. Because whenever someone asks me how do I define bioinformatics, so just tell them data science approach to biological sciences is bioinformatics and nothing more than that. Okay, so feel free to reach us anytime if you have any doubt or need any help in these fields. So we of course provide free of cost counseling services, and along with these, we also provide mental health related issues. Sorry, mental health counseling. Okay, so if you are facing some troubles in your academia or some other aspects of your life, feel free to contact us. We have a team of psychiatrists and psychologists associated with us. Extremely sorry. Along with that, we train the students in the field of bioinformatics and data sciences. And we are working on the aspect and trying to create internships with research institutes and few industries for those students who will be clearing all the different criteria and the parameters of our required courses. So we'll be able to provide you internships into industries as well as into research institutes. This will be started soon, and we are trying to build one of the India's biggest scientific community, because we have a large number of science background scientists, researchers, students associated with us. And with these motive, we try to conduct regular events which are totally free of cost, so that students and people from these fields get a good idea and seek experience from the experiences of experts in these fields. So this was our launch event, which was the first event of our Techmedbuddy, in which we had invited Dr. Swarnendra Singh, who is working as a senior head in industry, and he is placed in Thermo Fisher. We had Dr. Vatika Gupta. She is again working as a industry expert. Then we had Vasudev Singh, who is a research and development engineer in Mercedes Benz, and it was me. Then we conducted the second event with the motive of depression, the dark side of competition, where we had invited Dr. Hemika Agarwal, Purnima, and there were one more speaker, Dr. Rabia Khatun, where we covered the aspects of what is depression, how to tackle depression, and others. And we have uploaded this session of our YouTube channel. The quality of the recording is definitely not that good, but it's not that bad also. And past 2 weeks before, we had this particular event where we had Pawan Verma from Elucidator, who tried to give a brief idea about what is TCGA, what are the different applications of TCGA, and how these are being used in cancer research. And it's today's event with us having Dr. Kunwar Digvijay Singh, so who will be shedding light on today's event. And guys, one thing that I would like to tell in the beginning, that if any of you have any doubt, anything to say in between, please do not text. Feel free to unmute and raise your point in between. Okay, and it would also be great if you go to switch on your camera after the session, because we will be on the presentation mode, so it is of no use in switching on the cameras right now. So we'll be starting the session, yeah, fine, in 5 minutes. So now I give the stage to Dr. Kubar sir. So sir, you can start the session from your end by giving a brief introduction. And I feel that it would be good if before starting the session we could get an idea about the audience. If 3 to 4 students could just give their brief ideas. Yeah, over to you, sir.
That yeah, that's very important because I thought that it will be a general discussion between all of us, and there are people from different backgrounds. So if they are hardcore biological background, it will be much easier for them. If somebody is from different science background, they will need some little bit more idea about DNA, RNA to know how to use them or how we are using them for our clinical workflow. So yeah, just few of them, they can just let us know their background. And another important thing, if you have any question, any doubt, either you can just jump in between and you can ask that, or you can write down your question and at last we can discuss about that.
Sure, sir. So let me call few random people in between, and they would just give a brief introduction of themselves. Yeah. So hello, hi Anuradha, are you there? Hello. Yeah, Anuradha, am I audible to you? Might be she's not there. Yeah. Hello, hi Dinesh, sir. Hello, hi Dinesh. Hello. Hello.
Alok,
Yeah.
you had that form, right?
Yeah, yeah,
Google
yeah.
form where they may have mentioned
Yeah, fine, fine, fine. So I could give idea from that also. So let me share the screen once again. Is the screen on share more option only?
Yes.
Oh, great. So fine, sir. So
I assume most of them will be from biological background,
yeah, yeah, yeah. Almost all
but
of
there may be
fine.
few.
So, sir, we have master student with us, we have undergrad student also with us, and we
Mm,
have she's a graduation student only, and
okay.
so we have MSc, MTech, and PhD score, and undergrad definitely. So we have students from
Uh,
all hierarchy, and of course they are from the biological sciences background only, bioinformatics and
mm-hmm.
biological sciences related.
Yeah, that's fine. Like,
Fine. So you can
uh,
start then, like by giving your introduction, and then you can start. Thanks.
so hello everyone. I am Dr. Kunwar Digvijay Narayan, and I am working at University of Illinois at Chicago. My lab basically deals with use of DNA, RNA in clinical services. So what we do is we use these two molecules to identify some actionable variants, uh, particularly focused on the cancer. So we like we develop the assay and then we use them to identify some actionable variants that can be targeted by the drugs available in the market, or they are like clinical trials going on. So if there is no drug available for now, they can be like enrolled in the clinical trials and they can get benefited. So that's like my gist of the work, what we do here. And to know all these aspects, like why DNA RNA is important, I will be giving some brief idea how to use them and what we get at last and how they become important in nowadays, since this is the era of precision medicine. So you have like earlier, there was a time when people were getting a single drug for every kind of cancer. Just like if you have a fever, you are taking any paracetamol or similar drug. But cancer is a very diverse disease. Every tumor is a different, and then it varies between the person to person. So precision medicine basically became a reality when we have advances in the sequencing technology. So let's go there. I hope I will be able to give some brief idea, and then if that will make you more interesting, you can further, like I will be providing some links, you can go through them and then you can read much about it. And anytime, since Alok said you can reach to us, so feel free to ask question or reach out anytime you want. So let me share my screen.
Yes, sir. Now you can share.
Okay. So I guess you guys are able to see it, right?
Right now, not so.
Uh, how to do it?
एक second.
I guess there's something needs to be done from your side.
You are not
Alok,
able to share the screen or what?
it's showing me, but
Guys, anyone please
I
confirm is the screen visible to you?
Not yet.
Not yet, sir.
okay. There was a like simple way to sharing. I am more comfortable with Zoom, you know.
Yeah, I
Uh,
agree. Like in the bottom, over the call button, you had one option, present now. Yeah,
yeah,
now it's
I
visible.
guess we are getting there.
Yes, yes. Now it's visible.
So
Yeah,
it's
great.
visible now.
Yes, yes, sir.
Okay. So
So you can bring it to the presentation mode, sir. Like,
yeah, you know, sometime. Okay. So
fine.
let me get the pointer. Okay. So as I said in the beginning, we are using DNA, RNA for our clinical services. That's providing services to cancer patients who are coming to our hospitals. And right now, we are just focusing on the patient cohort which are coming to our hospital. But in future, we will be expanding this service to the other hospitals located into the Illinois. And we used to get like sometime our lab is also a referral lab. So if there is some complicated cases, they send us the sample and then we work on it. So DNA is known for a very long period of time. Uh, Hey, look, is there an option I can see you guys while I'm
No, sir. No, sir.
presenting?
No, sir. In this, there is no option.
Okay. So I don't know like how things are going on, but that's fine. Uh, so like DNA was discovered a long ago, but at that point of time, people didn't realize how it will revolutionize our everyday life. Everything is encoded in DNA, and that's get like translated into the RNA, and then you have a workforce of protein. Uh, so, like moving forward, um, I will be covering basically these three topics. So, what is genetic material? Their function, if they get some changes, what will be the consequences? And then, how do you know about like if there's any change? So, we need to sequence them. So, there were like three generations of sequencing. The first one was Sanger sequencing, and that get evolved further. Now we are having NGS, and there's the third generation sequencing technology, which basically focus on long reads. So you have Oxford Nanopore and PacBio that gives you really a very large strings of DNA sequence, maybe around 100 kb or something like that. But they all have their pros and cons, and that I will be like showing up in next few slides. And the question is, you have all the information, but what are their utility? That's the most important thing you should know. So, like if you have different kind of information, but if you don't know how to use them, then there is those information are not worthful, right? Uh, So basically, these became important because we are able to use those information to diagnose a patient, or even they are of prognostic value, meaning that how the disease will move further in future, that can be predicted, but it's not absolute thing that you should also know. So coming to the DNA, like we, since you all guys are from DNA or like biological background, you know, but it's just a recap. So we have like four bases, which is giving like 3 billion base pairs of DNA sequence in our body, and that's making like 23 pairs of chromosome. Uh, DNA get translated like we know the central dogma where DNA gets transcribed into mRNA, and then you get a protein, which is the basic workforce of the body. You need to perform some function, your protein is working there. So if everything is working fine, like you have no issue, they are perfect machinery helping them to divide the cells, control the cells, you have no issue. But when you have system fault, like you have a mismatch repair system defect, or there are some certain mutations, just to note down, mutation is not always a bad thing. Sometimes it makes you like better fit for the environment, but there are cases where things like change, there are substitution and other things that change the base pair, and that leads to the malfunctioning of mRNA and ultimately leading to the protein. If you have a bad protein, you are going to have problems. and those problems, like one of them is cancer. So how, like, okay, there are system which can control entire stuff, but there are instances where you have a single base substitution, or like for example in first panel you see the single base substitution, and that changing the amino acid histidine to proline. So obviously your protein doesn't need proline, but if you have it, there will be like malfunctioned or misfolded protein. So there are like varieties of ways where can change in the human system. So they are classified as a like missense mutations. So you are like getting changed amino acid. They are nonsense, which is leading to the uh, a stop codon, and your protein is like early terminated. You don't have the entire component of proteins with you. So again, it's a different protein. There are instances where like few bases can get inserted into the sequence, and if that is not getting corrected, you have different sets of like amino acids here. Again, that's leading to the malfunction protein. So there are like varieties of ways. There are like translocations, like in this case, some portion of your chromosome is moving to other chromosome and then giving to a different complex of protein. This is example of BCR ABL, where like a portion is moving from chromosome 9 to chromosome 22. That's leading to Philadelphia chromosome, and this happens mostly in like CML, chronic myeloid leukemia. But since we know these translocations, and fortunately there is a drug available that can be used to treat this kind of patient. So this was just like, you might be thinking, if there are the system that can fix this problem, then why we are having mutations? But your proteins get malfunctioned like these events, and cancer is basically related to somatic variants. So this happens over a period of time. It's not today there was still dividing and there was some problem and that didn't get corrected. You have various exposures, lifestyle problem, and then that leading continuously to the development of cancer. So you know, like you now have an idea how can changes occur in DNA, but how do you know about it? And there sequencing comes into the picture. So sequencing is nothing but to know the order of bases in DNA, RNA, and there was like guy, uh, Frederick Sanger, who developed the Sanger sequencing. We are like talking about Sanger sequencing mostly because this is the most widely used technique even nowadays, and that considered as a gold standard. I will come to the gold standard thing in few minutes. Why we call it gold standard? So there was like time frame. Basically, the time has been divided based on the what was coming out from the sequencing. So like nine, up to like 2000, the Sanger sequencing was the primary sequencing method that was used even to complete the Human Genome Project. And then the problem is with the Sanger sequencing is it you can work one gene at a time. So or even one stretch of DNA at a time. But you imagine a 3 billion base pairs of DNA sequence. If one tries to use Sanger sequencing to finish that, it will take forever. And how much money you have to spend, you cannot imagine. That's why, like, when Human Genome Project was formed, there were like various groups working across the world to get it finished, and it was a really huge investment from the government of America. So to overcome this issue, like you can look at one gene at a time, people will started working if it is possible to do a parallel sequencing, like at the same time, if you can look at millions of leads, millions of leads can be like generated and they can be sequenced. And that's the most benefit of NGS. But like you have another issue with NGS. You don't have expertise. Like you always need a bioinformatician or computational background people to analyze all those data which is coming. It is just like ATCG coming out and how to interpret those. So, like from the biological background people, it's really a challenging work to understand those data. So, but that's the like lagging part of NGS. But now system is evolving and people are learning it. So now it's easy to interpret the data. And then since the small gates assembly things is a yeah, actually these things are creating a whole lot of problem. And you have a time where you need like genome assembly thing. So there you need long stretch of DNA to complete the entire genome. And there the third generation sequencing coming into the picture. If you look at the application part, sequencing is involved from the before the birth of the people to the end of the people. So if somebody like planning a baby, the parents can go for genetic testing to see if there are any recessive alleles are there that can cause some sort of problem in the future. In upcoming baby, they can like identify it and they can like the physician can try to solve the problem or there are like various ways they can get it fixed. So like a fetus is born, you have a newborn baby and in the beginning there is no disease like it may be disease is there or maybe not there. If it is there, if it is not symptomatic, you don't know if there is a disease. But if you are performing sequencing at this point of time, you can know if there is any somatic variation or not that can lead to disease. If you found something that okay, these are the variants present there, and if any collective measure available, either drug or some treatment, you can try to like prevent or you can start having like physical activity. Like nowadays we hear about diabetes, polycystic ovary syndrome in females. These are like lifestyle disorders. So if you know, okay, you have these kind of variants there, you can start working on it. Uh, so like over the period of time, there are somatic mutations getting accumulated into the body. If there is any cancer, that can be screened. You have some treatment available for that. You can use that treatment, try to like treat it. That will like enhance the lifespan. And you are having like instead of 70 years or 60 years, you are living up to 90 plus, and then you die. Even then, your DNA can be taken out and it can be analyzed if something went wrong or not. So you see the whole spectrum of application of like sequencing technology. Now there are like question what kind of sequencing options one can use. So if somebody wants to study the whole like entire genome of the system, they can go for the whole genome sequencing, but it's pretty costlier affair. Needs lots of expertise from the people. You really need a lot of people to work on it. Uh, but there's another option, targeted sequencing. So targeted sequencing come into the picture mostly for the cancer patient because there are certain genes uh, which is known into involved into the cell growth and growth maintenance. So there are signature sequences that can be identified, and if there is any variant present, and if they are actionable variants, meaning that if you have any drug available to treat that one, then you go for targeted sequencing. The other option of doing instead of whole genome, you can go for genome sequencing. That's your entire coding sequences present in the genome. That's also sheds some lights like normal protein and abnormal protein. Uh, so that's basically genome sequencing to sequence the coding regions. Another option is RNA-seq. So in RNA-seq, basically you work on entire transcriptome. So basically mRNA. Since it's the expression of RNA that leading to the different kind of proteins, and there are like regulatory aspect of it. So if you have entire transcriptome, you can predict uh, like where things went wrong, or some proteins are like some mRNAs getting overexpressed, or some are under expressed. You can compare between two sets of like uh, population or two sets of patient and see the differences. And recently we have single cell where you can look at single cell molecule. You can get the DNA, RNA out of it, and you can try to see how each cell is behaving. This is becoming more important now because there is a tumor heterogeneity in the cancer. So, like you have a whole bunch of cells living together, but each cell is behaving differently. Uh, so, in that case, single cell becomes pretty important to do. But again, expertise is needed. Uh, is everything following me up, or because they are like complete silence? I'm wondering if I should move forward, or if you have any question till now, you can ask.
Yeah. So guys, if any of you have any question in between, feel free to raise your point here.
I
Okay.
think no one has any question.
Yeah, sir. Sir.
Yeah, if I'm moving too fast, you can let me know, or too slow. But yeah, just let me know, okay?
Sure, sir.
So now come to the Sanger sequencing, which was like basic workhorse for Human Genome Project. So it's a pretty simple method where you use when you use Sanger sequencing, when you have less number of genes to study. Suppose you need to study 4 or 5 genes. Uh, so you can design the primer, and then you perform Sanger sequencing. You use like the 4D deoxynucleotide present that when get. I hope you guys understand PCR. Uh, so you do the PCR with these 4 deoxynucleotide, and that leads to generation of multiple fragments of DNA. And when you run it on the capillary, it's a kind of capillary electrophoresis. We used to run agarose gel, but in this case, you need a page polyacrylamide gel system, and it has a capacity to resolve one base pair. Like there is, you see increment of one base pair when you see the graph, and you get like fluorescence signal that get translated into the bases, and there are like software to call these bases. Then you manually visualize if peaks are perfectly fine or not, or there are some slippage. You can rule that out. So the pros with this Sanger solution is you can have a read of up to 800, 900 base pairs, but it's a time consuming and cumbersome process. If suppose you need to study 100 genes, with different like combinations, it will take whole bunch of time to perform that process. But the best thing is whatever observation you get from Sanger sequencing, that's like 100% correct. Uh, so that's why we call it like gold standards. And even nowadays, it's in application, and I guess and something advanced come out from these, it will be remain there. Even if you see something in NGS, like you did NGS for 100 or 500 genes, and you see the variants, uh, the the pros is with NGS, if you have like millions of leads for one strand, that's like confirmatory. But in order to validate that, you need to design a primer, perform the central sequencing, and analyze the data to see if that variant is correct or not. So
Sir, I have a
the
point here. Hello. Yeah,
yeah, yeah, yes, sir.
so it means that like Sanger sequencing is one of the like most important technique if you want to confirm the results of our variations, right?
Right. So the thing, you don't do that every time. Suppose you have a set of data where you're seeing, like for EGFR, EGFR is the most common mutant, like driver mutation in lung cancer.
Right.
But if you see like EGFR in some other type of cancer with the same variant, and there are like less number of leads, then either you perform the NGS again to confirm that, or you can like design a primer for that, like the coding region, and then you do the Sanger sequencing. So Sanger sequencing will give you an absolute idea. If it is present there, it will show up.
We got it.
That's the power of Sanger sequencing, and that's why like applied biosystem is continuously working on it, and it's not going to get replaced.
Right, because when we perform the NGS, like it has the more higher level of mistakes probability at every particular
Yeah.
place. Right.
Mhm. So yeah, I will come to that too.
Sure.
They are like, we know NGS. You do NGS and you get entire information. That's not true. Obviously, you have to substantiate your data with like other information available in the market. So they are like, even VDSU also is there. They are like different um, platform available, basically Illumina, and then you have iron semiconductor from the applied biosystem, different technology, and there are like different limitations. But yeah, they are still existing in the market, and they are improving so much. We really rely on them to perform our day-to-day work.
Right.
So I like kind of summarize when what are the outcomes when you do the single sequencing or NGS, and when you go for single sequencing or NGS. So single sequencing, obviously it's a fast. You have some doubt about few genes, or you want to look into some genes, you design the primer, you do the PCR, and you run the instrument, and you get the data. But it has like low sensitivity, meaning that your allele frequency should be at least 15 to at the 15 or 22%. That's the detection limit. If you have below that, your single sequencing won't be able to identify that variance. So it obviously, if you look further, it's have a low discovery power. Like if you want to look at multiple genes at a time, you won't be able to do that. And also scalability issue is there. Like at a time, there are like 96 capillary available in the markets. So at a time, you can run 96 samples at a time. Like you have to do multiple reactions, and they are like time consuming. Money is also a matter. So if you have few genes, obviously you should go for single sequencing. But if you need a broad picture, like multiple genes at a time and low cost input, obviously you should go for the NGS. Uh, but it's not applicable when you have less number of targets to look for. Uh, those is even it can discover the variance even around 1%, although they say it 1%, but at least in our experience, it should be around 5%. Sometimes we were able to get around 2% or even 1%, but if it is like around 5%, it would be easier to identify there. So those the pros and cons for NGS. Now coming to the NGS steps, so there are like different ways of doing it. It depends upon your need, what you are looking for, and how accurate data you want, and how much time you have. So they are like hybridization or amplicon based. So in hybridization, you design some targets and you like get your DNA, RNA from your sample. You do some PCR and then you hybridize the molecules. They are complementary to each other. They makes a double stranded molecule and then you pull it out using beads. In amplicon based, you design like multiplex primer, various sets of primer that can be the PCR can be done in a single tube, and then you identify those targets. So that's the basic difference between two techniques. There are like fragmentation step involved. This is when you have like RNA sick. So uh, usually nowadays we get various types of samples. You have phase progen samples and then you have FFPE samples. So phase progen means you draw blood or you get some biopsy. You immediately get the DNA, RNA out of it. So they are like more intact. But if you need to do the biopsy and that's that needs to be histologically studied. So you do the ISC or histology that needs to be reviewed by pathologists to see the tumor content. But that has another disadvantage. Although the FFP slides can be saved for longer time, and suppose if he is so in real world, you see, we get a patient that was diagnosed by like some cancer, but clinician is not sure, so he asked for surgery. So the guy or the person gets biopsied, and their tissue get fixed like formalin fixed on the slide, and you perform a staining. HNE is a staining on that. Then you see the tumor cells on the slide. That tells you how much the tumor content is there and whether NGS can be performed or not. So this all takes time, right? So you don't want to get your tissue wasted. So that's the FFP things comes into the picture. And once like pathologists review, they confirm, okay, this much of tumor cells are present there, then you move forward to get the slide dissected, get the DNA, RNA out of it, and then you go for NGS. So capture amplification, this is both like similar to hybridization or amplicon based. Once you have your DNA, RNA available with you, you go with library pair where you like add molecular barcodes to it, and then you choose a sequencing platform, whether it's Illumina or Iron Torrent. You perform the sequencing, and you have a whole lot of data with you which needs to be aligned to a reference genome, and then you perform the variant calling. Uh, these are all like alignment and variant calling things are bioinformatician job. So they use some algorithm, they refine the algorithm, and then they come with like, okay, there are 300 or 500 or 1,000 variants present in this particular sample. So that's a kind of NGS in nutshell. If I am showing here, uh, since Alok basically talks about RNA-seq, so there is like how to do RNA-seq. You
Pardon,
can
sir.
do,
Hello.
yeah.
Sir,
Mm-hmm.
in the last previous slide, can you please focus once again on amplicon sequencing?
Okay, so
Yeah.
yeah. So Alok, the thing is in amplicon sequencing, you have like a stretch of DNA, and then you design like there are like long genes available, and there are various codons present in the DNA for a single gene. So you target a small region, okay, to make a primer, and then you perform the multiplex PCR. So for various codons, you have various fragments available. It's all PCR based instead of hybridization. So here, getting a false positive chances are more
Okay.
because over the period of time, your polymerase gets exhausted. There the confirmation in structure. So those things happens that cannot be ruled out. In hybridization, it's more precise. Okay, you already designed a bit and you have a target sequence there, right? So when they are like complementary, only they can bind, and then you pull out the complementary one out, which exactly your targets. So, but the con side is on in hybridization, you really need lots of reagent, lots of expertise, and it's a time consuming, and that's why it becomes costlier. So until unless that not necessary, people don't go for it. Basically, people go with the amplicon based. It's faster. For example, if you do the RNA-seq with like Agilent RNA-seq library prep method, it takes you at least 3 days and followed by sequencing. That's like overnight. So like it takes 4 days. But if you go with the amplicon, you get the DNA RNA, you perform the PCR, your templates are ready, you set it on the chip, uh, and next day you have results in your hand.
Got it, sir.
But but it's a scalability issue also. If you are doing a large sample, then like for Illumina, you have high sec and Nova sec that can multiplex like you multiplex multiple samples at a time, and you have like millions of reads out of that. So it's a context based. For diagnostic purposes, you really need a faster collect results, of course, and turnaround time should be like very less, meaning that you should get the results as soon as possible so that like treatment can be started at earliest stage. So all the techniques are perfectly fine. You cannot say this one is good or that one is bad, but it depends upon what you are looking for, and then accordingly you plan your entire experiment.
Right.
Uh, so this is just an example like how a RNA-seq workflow goes on. So as I was saying, this is hybridization based protocol. So you go to the genomic location, like what you are looking for, then you design the targets that will be used as a like to hybridize your target molecule. Now other side you have I don't know if I said about the pros and cons of FFP and phase frozen. So in phase frozen samples, you have intact molecule. Uh, there are like no chemical modification or something has happened, so you get like very nice DNA RNA. But in FFP, since there are formalin involved and there are various processing stages involved, that sometimes introduces artifacts like change the bases or even you get low quality DNA RNA. But we at this point of time basically we deal with the FFP because you can save your samples for a longer time. And like the sample moves from one place to another place to get it reviewed by the pathologist. It's getting processed somewhere else to get DNA, RNA out. So raw samples, you cannot do those with the raw samples, like when you primarily get the sample out of the patient. So if you have like intact RNA, that needs to be fragmented because there's limitation of reading the base pairs by each instrument. So NGS basically sequence gives the reads about 200 base pair or maximum up to 300 base pair. So you want to keep that size intact. Like your fragments should be in that range to get the prepared sequencing results. You quantify your RNA, like there are certain requirements how much RNA needs to be used for each protocol. Sometimes it needs 2 to 3 microgram, sometimes 100 to 200 nanogram also works. So there are like various categories of RNA-seq method. You fragment them, um, and then you ligate some adapter so that like your fragment of interest is present in between those barcodes, and you like another important aspect. Uh, so in RNA-seq or transcriptome study, you don't study RNA in its native form that ultimately needs to be get converted into the DNA. So every sequencing technology reads only DNA strands, not RNA. And that's why to do the RNA-seq, at some point of time, you have to make cDNA. That's the complementary DNA of that particular RNA. Okay. So you get cDNA library out of it, and then you use the bits you design, like your target sequence, and then you hybridize them. And you do it for overnight uh, at 65 degree, or sometime they have faster protocol where you can do it for 4 hours. Um, the captured molecules you pull out using beads. That's a streptavidine beads. So that pulls out only your hybridized molecule. Okay, so non-specificity is extremely low here. Then you amplify your target molecules, like if you have 10 or 15 copies, you do the PCR, and that's get number to like 1,000 or 5,000 uh, molecules. That comes like a finally prepared library. So the finally prepared library looks like this. You have your target of interest here. Then you put some index to identify, okay, this sample, like how to identify your samples. That's indexing that's provided by the organization. And you have like flanking regions. These are the regions which goes on the flow cell, uh, where it binds to the, like they are the adapters present on the flow cell. I don't know if this is visualized. I can increase a little bit.
Sir, you can bring it to the presentation mode, then it will be visible.
Okay.
Yeah,
Yeah.
now a little bit, yeah.
So this entire uh, entire option. Okay. So when you do entire things, you get this. Okay. This is a nutshell, a library in a nutshell. You have your target sequence, then you have sample identifier, and these are the Illumina, like which platform you are going to use. They have their own barcodes that needs to be present there. And these like yellow flanking regions, they are also 8 base pair or 16 base pair sequences that when you put it on the flow cell, it binds to the flow cell through these sequences, and then you get like so this one can be summarized here. You extract DNA, RNA, then you perform library, you add the barcodes there, and these library, once quantified, they go on the flow cell. So flow cell is nothing, it's a glass slide kind of thing, which Illumina uses. Like there are various barcodes present over there. When you put your molecule, it gets bind there, and then it's get amplified. Another round of like PCR happens here too, where your molecules goes inside, binds there, then it gets amplified, then it forms a bridge. There's another like other side of molecule, and then it gets amplified again. So basically, at last, you will be getting two reads for one molecule, and that's why like read one and read two. You perform the sequencing, excuse me, and you get sorry, you get FASTQ file. That's get aligned to the reference genome, and then you get PAM files, and then you further work downstream to get variant call files where your variants are present. And Alok will be covering like these entire workflow. Uh, right, Alok?
Yes, sir. Yes, sir. Yes, sir. Is it done from your
Okay,
side, sir?
so sorry, Alok.
Is it done from your side? Like these many slides we had to discuss.
Uh, there are like few. Just I can go a little bit faster if you want.
Sure.
Uh, so that was
Yeah, it will be
like
good.
Illumina.
Okay, it will be good if you cover the coming slides also because Iron Torrent is a very like famous one nowadays.
Yeah. So when you like in our case, we usually perform RNA-seq when we are doing research stuff, like we want to discover something new. But if we want to perform some clinical services, then we do the iron to end sequencing. It's much faster, easy to perform, and like it's less costlier too. So in this case, you get your DNA or RNA. If RNA, just reverse transcribe it. So you have cDNA with you. You add primers base, amplify the targets. You have amplicon where some barcodes are present. Basically, this will be used to adapt, uh, ligate the adapters. You partially digest it, and then you have blunted ligation where you add some barcodes. So these barcodes, sequencer doesn't know what you are going to give it, but it will only recognize these adapters present on there, and then it goes on a chip. So this is the Maxwell instrument we use where you put your DNA RNA. It will finally give you the DNA RNA. You put your samples, you get DNA RNA out of it. You use a fluorescence detector to quantify your DNA RNA. It's pretty precise. That's why fluorescence things comes into the picture. Otherwise, earlier we used to do a spectrophotometer to quantify your DNA RNA. You are like getting some ratio 260 by 280 to see the purity and the stuff. So then you purify your library using the agent called impure bits. You can play around with the concentration to optimize it to get a fixed range of your targets, like size of the targets. Uh, we use tape station, Agilent tape station. So it's a fancy gel, you can say, but it will give you whether your library falls in the range or not. If it is oversized, then you have problem. If it is undersized, you have problem. So technically, the expected size range is around 200 base pairs. So you see the average size is like 198 or 200 base pair. So this tells you a perfect library made. Uh, Hold on. Okay. So once you have library, there are like various way of quantifying it. You need certain concentration to proceed further. And in iron-to-iron sequencing system, uh, it range what kind of chip. So this is a semiconductor chip. It has its own capacity, and depending upon your number of samples and how much reads you want, you like the multiplexing changes. You can use 4 library, 8 library, 16 library, but that's all depends upon your requirement. So to quantify the library, you need to perform qPCR. You get certain values, then you dilute it to get up to like if we are using iron 540 chips, we usually do the 50 picomolar dilutions, and you can sequence up to 16 library. So in this case, you have both DNA RNA since it's a clinical service. So you need to have both DNA RNA available with you, and you make library both of them, and you also put a reference standards. Reference standards are standards where the variant allele frequency is known, the variant is known. So that tells you if your library prep and sequencing went perfectly fine or there are some deviations. And you know these needs lots of validation things, like you do multiple times cross platform validation to know if your system is working perfectly fine, or if you observe sometime there are deviation, but how much is deviation, whether it's acceptable or not. So those are the clinical sites. Okay, you just cannot randomly say, okay, I don't want to use reference standards, and my since my sequencing workflow is perfectly fine. You cannot do with the clinical settings. So you do all those things. Then you this is the IronSafe machine. People earlier used to load the chips manually, but that's not getting appropriately loaded. Like sometime underloading, sometime overloading. So you don't get appropriate data out of it. Then you put the chip on the sequencer. This will go for like 3 hours, and then it automatically there are like current suite software which aligns your sequencing reads to the reference sequence, and then there is another software that can give you the VCF out of it, and then you can look into the data by yourself. So that's the iron-to-iron sequencing in itself. Uh, I also put a slide for Oxford nanopore, but this is not like something I do in my lab. But this is still under developmental stage, because in this one you have a like protein molecule embedded into the membrane. You pass single stranded DNA through it, and there are like fluctuations in the current, and it's like getting interpretive what kind of graphs you are getting out of it, and then you determine the base pair. It's good to have this one when you really need longer stretch of DNA read, but for clinical applications, these are not applicable, and it's costlier. Also, it's hard to interpret the data. So they are still working on it, and hopefully in future there will be more advanced version out of it. But I am not much familiar about it, so I cannot say much. Now coming to the cancer diagnostic part, so there are like various ways of doing this diagnostic workflow, but in this case I am showing one of them. This is part of targeted sequencing, where you know what are the genes involved in the cancer progression or maintenance. So organization like it was developed over a period of time. People were doing research, uh, and they found that okay, these are the variants which involved in the cancer development or cancer progression. So they classified it, and there are like different sites like NCI, NCCN, FDA, all they are like gathering these informations, and the company identify those targets region. They design the primers. Even we can also do that, but if it is already existing in the market, you don't want to waste your time. Uh, but if somebody is in the discovery lab, like research lab, they can work with the mouse model, human model, or whatever possible to get new information out of it. Uh, so these are some of the like driver mutations. Driver mutations meaning that they are essential to help the cell grow or maintain them. Uh, and they can be like classified like they are not every variant is pathogenic, and this is one of the interesting paper. If somebody is interested, you can read it. So basically, there are like four variant categories has been defined, and depends whether they falls into tier 1, tier 2, or tier 3 or tier 4. So tier 4 basically these are the benign variants, means if they are there, they are not leading to any cancer type, or at this point of time we there's no evidence that they are involved in any disease. They are like normal variants. But there are other three classes of variants in which you have level A, level B, level C, level D. So here you have FDA approved drugs available. So if something you find out is a tier 1 variant and what level of that variant is, so that will tell you if that patient can be get treated with some drug or not. So I am showing one of result out of our lab. Like we have these like thousands of results, but this is a scenario where a person was found to have melanoma. So melanoma is nothing but a skin cancer. Uh, and his or her, we cannot tell about the patient ID. So uh, DNA was used, and it was used to make a NGS library for end-to-end sequencing system. And after doing sequencing, it was found that it has like three types of variants present in there. So tier 1 variants in the level A. So it's like therapeutic, meaning that you have a drug available to treat this patient. And then you have tier 2 variants, and there are like various categories involved here. You also get tier 3 variants in which you can see the amplification, like few genes got amplification, meaning that you got multiple copies of it. So obviously instead of having one copy, you have multiple copies. Those proteins are getting overproduced, and obviously that will drive the more cell divisions and cell maintenance, right? So to summarize this, for NRS, we have a drug available, and there are other clinical trials going on. But you know, you might be thinking, okay, this person has NRAS mutation and it can get treated with this one only. So that's not truth also, since it has other driver mutation too, and I just describing few of them to let you know cancer is a very complicated disease, and it depends like how your disease is progressing depending upon the what kind of variants you have. Sometimes you have sensitive like if you have a variant and that's sensitive to a drug, that can be treated with that drug. But there are instances where you have two or three different variants present in the same person or same tumor type, but that will not respond to one drug. So it needs to be like a studied what kind of combinations you have and what kind of drug treatment can be used. So, like, but still, if you look at 10, 20 years before this date, there was not so much information we had. But now, since we are getting these informations out of sequencing, so we are able to manage like more and more patients. Um, that's all from my side. I put some like website address. If somebody is interested in learning more, they can go through it.
Yeah. Fine. Yeah, thanks.
Yeah.
So, like before starting my topic, I would like to have the questions. So, guys, if any of you have any question till here or any doubts or anything that you have in your mind, feel free to ask here. Okay, because one of the most important point for which we are trying to conduct these type of sessions is not to give you the knowledge. Let me tell you frankly, not to give the knowledge, because in 2 hours of time you are not able, you will not be able to fetch everything. It is not possible at all. So we are just trying to build a networking so that you could know people from different parts of the world, different parts of the India, and different places, so that in future, if any point of time you struck somewhere, you would have in your mind that yeah, I know that other person, he can help me out there. So, and of course, like by taking these type of sessions, they help to brainstorm you and to put emphasis on your mind to think in a different aspect. Okay, because let me tell you frankly, like right now I have got to know 3 to 4 different aspects. Like I wasn't aware like how this NGS is being used into the real world application, that is like the tier 1, tier 2, tier 3 applications. And by getting the type of mutation, we are able to get the type of the drug also, that which particular drug can be used, which is FDA approved. So these type of sessions are just to help you make connections and to introduce yourself to the people and get a network. Okay, so if you have any type of question or anything to your head right now, feel free to ask that. We can give 2 minutes of time here for the questioning session, and after that I will tell you how because these were at the molecular levels, now I will be telling you how how we analyze the data that we get from here. Okay, and I'll be covering only the sequencing data. I'll be covering the sequencing data, that how do we process with the gene expression matrix data. Okay, so now over to you. Like if any of you have any questions, feel free to ask, please. It is not possible that you don't have any questions, let me tell you frankly.
Yeah, the question can be anything.
Yes.
It's not like so. For example, I should say it's not always possible to have a drug available in the market, but there are like clinical trials going on across the country. Like I know for here for sure. In India, is it's also happening there. But suppose if drug is not available, but for the particular variants, if somewhere is clinical trials is going on, patient can get enrolled there, and if that benefits him or her, that's really nice, right?
Definitely so. So, guys, anyone having any questions?
I don't think.
Okay. So, Ekta, do you have any questions? Yeah,
So,
Kruti. Yeah, hi.
I was just thinking, like, is it possible to sequence the baby, like, who is present in the womb of a mother?
Yeah, that's like a fluid can be taken out from there, and it's like it depends at what stage it is. Like I guess it's after 1 month of pregnancy or 2 months of pregnancy. I'm not so sure, but it's applicable.
I guess it's 3.5 or 4 months, I guess.
Yeah, I didn't like.
Yes, yeah,
I'm not so sure, but it's quite possible.
right. Because this technique is called amniocentesis. Yeah,
Right,
this concept is called amniocentesis, in which what we do basically is that we take out the amniotic fluid from the baby, like not the baby, basically the fetus, and then that particular fluid is being sequenced, and we are able to detect any type of genetic mutations or different type of things
right.
that is possible. And these are the things that is to be done in the time of genetic counseling, or a genetic scientist or expert always helps or recommends people when the couple have any chances of having any mutations or something in the baby, and these are very highly
Yeah, that's
recommended.
yeah, that's like before the planning a baby, you go for the autosomal recessive screening things. So there are like panels of genes involved, and that will tell you once you have that, you will know whether you are going to have any like downstream things in your baby or not. Um,
Right.
yeah, so that's doable.
Definitely. So I would like to pitch an example here. You know that albinism is a disease which is autosomal recessive. Okay, so like I have one of my friend. Okay, so his father is suffering. His father is a patient of albinism. His mother is normal, and then is not having the symptoms of albinism. Now, when I and he's pursuing MBBS, he's an MBBS student, and right now he has completed his MBBS. So whenever I talk to him, he always says that कुछ भी हो जाए शादी से पहले तो भैया चेकिंग पक्का करवानी है. ठीक है. So these things are highly recommended whenever you know that in our family history we have some type of disease or something, you should always go for a genetic counseling or something. Okay.
Yeah, it's good to have those.
Yeah. Kruti, I think we have answered the question.
Yeah, thank you so much for the insightful information.
Welcome.
No
Anyone
problem.
have any further questions? चलो ग्रेट या फिर ना लाइक एक काम करते मैं अपना सेशन स्टार्ट करता बिकॉज आई विल टेक हार्डली 10 minutes एंड आई विल रैप अप इन 10 minutes because I am going to just
Okay.
talking about the applications, how these things are being done at the analysis level.
So I'm closing my presentation. Okay.
Yeah, sir, you can close your presentation, but be in the call.
Okay. Yeah, yeah.
So,
I believe.
so please confirm is the screen visible, sir?
Yeah, yours is. Yeah,
So,
I can see that.
fine. Yeah, guys. So before starting with this plot, so first of all, I would like to give you a little bit idea about the data. Okay. So after completing the sequencing and everything, what we get in our hand is basically the reads, which sir was talking about previously. That we get the few reads, and then from those reads we get the calculations. We do the calculations, and we get a level of the measures that which particular gene is expressing at what level in a particular patient. Okay, and the matrix summarizing the expression of the complete set of genes in a particular patient is known as gene expression matrix. So gene expression matrix is nothing but simply the complete the complete set of expression values of all the genes in a particular human, in a particular patient. And when we have the data for a large number of patients, when we have the data for a large number of patients, of course this will be a 2D matrix then, because now we have the large number of patients. So we have samples which will be the patients, and and over the columns we have the features which are the genes. So this is known as the gene expression matrix, where we have a large number of gene values, and this help us to know when we will be trying to perform the analysis and everything, we will be able. Fine. So sorry. Yeah. So when we try to analyze these things, our motive is very simple. That is, we just try to find out how a particular phenotypic condition is different from another phenotypic condition. Okay. For example, suppose that I am a patient of cancer and you are a healthy patient. So what I will try to find out is that in cancerous state, what are the different set of genes which is driving cancer or making a person from healthy to the cancer, and the complete process of analyzing this thing, where we are trying to find out what are the different set of genes which are expressing differently in cancer as compared to control, is known as differential expression analysis. So this is the meaning of differential expression analysis, where we are trying to find out what are the set of genes which are expressing differently from the given set of condition. If we are comparing cancer with healthy, then we will be reporting that a particular gene X is expressing differently as compared to healthy. Yeah. So this is the analysis. This is the result of the after performing DEA, which is known as differential expression analysis of the genes. So suppose that we have 20,000 genes. Suppose that, because exact number of genes nobody knows. Let me tell you, because this number keeps on varying. Okay. So suppose that if we are having in our data 20,000 genes, so we will be trying to find out what are the set of genes which are differently expressed, and then we we are returned with this type of analysis, which is showing that over the x-axis we have log fc values, over the y-axis we have the p values. So p values is telling you the level of significance, that the amount of differentially expressed gene that you have got is statistically significant or not, because whatever result you have got, it is from the data, but is that data statistically significant or not, that you will have to tell, and that is being given by the p values. And over the x-axis we have log fold change. So these black or like gray colored genes is not differentially expressed. Like you can see here that these genes are expressing different from zero. It means that they are differentially expressed, but these are not statistically significant. Okay. Similarly, these red colored genes, these are above zero cutoff, so these are positive, these are having positive log fold change values, and they also have a statistical significant value. So we say these type of genes which have positive log fold change value and statistically significant, known as upregulated genes. Similarly, these blue colored genes which are present over the having the minus log fold change value and statistically significant, these will be known as down-regulated genes. Now the point is, हमने सेट ऑफ जीन्स लिए, हमने डिफरेंशियल एक्सप्रेशन एनालिसिस किया, हमें मिल गए कि ये वाले सेट ऑफ जीन्स हमारे स्टैटिस्टिकली सिग्निफिकेंट है और डिफरेंशियली एक्सप्रेस्ड है पर्टिकुलर गिवन स्टेज में. नो व्हाट टू डू आफ्टर दिस, अब इसका क्या करें हम. ओके, सो आफ्टर दिस फर्स्ट ऑफ ऑल you will be trying to find out how these particular genes are trying to help us predict a particular patient is cancerous or not, and there we go with the machine learning based models. Okay, so when we try to find out the set of genes which are differentially expressed, you can build ML classifiers which will tell you that based upon these set of genes a particular patient is cancerous or not. But before that event, based upon these set of genes which are statistically differentially expressed, we try to perform dimensionality reduction. So if you guys are not aware of these statistical terms, because these are machine learning terms, okay, so that is the matter here right now. So after getting these genes, based upon the expression value of these genes, we try to plot these samples. So here you will clearly see that these green colored dots represent the control patients. It means they are healthy patients, and these red colored dots are representing your cancerous patients. And here you can clearly see that the control group is present as a cluster and the cancerous group is present as another cluster. Okay, so and this is definitely your desired result also, that we should be able to find out those genes which are able to differentiate a patient from cancerous or to healthy. Yeah, fine. And after getting these set of genes, we also try to find out that how these genes are interacting with each other. So suppose that we take the complete set of positive upregulated set of genes, and then we will be trying to find out how those set of genes are interacting with each other. And this network of interactome, the network of interaction of the genes, is known as PPI network. So this means that protein-protein interaction network. So sorry, because we know that genes codes for proteins. So we are trying to look at the interaction of these proteins, that how are these proteins interacting with each other. Okay, and after getting these PPI networks, we try to predict that, yeah, like suppose that here this particular gene, this gene is known as a hub gene. So hub is nothing but simply it is that particular node will share the maximum number of degrees. Okay, so here if I am telling that PGK1 is a very important gene, okay, and this particular gene is sharing its network with these many adjacent genes, then we will try to find out, okay, so these complete set of genes, what are the pathways in which these particular set of genes are being included. And after getting the set of pathways, suppose that all these are the set of genes obtained from positively upregulated gene from cancerous against control, then we'll say that, yeah, these particular set of proteins are included in, suppose that these are included in blood circulation, just for example, it is just an example. Okay, these particular set of genes are include is responsible for coding or leading to the pathway of glycolysis. Then we'll say that glycolysis is being updriven in cancer. This is just an example, analogy, not the real example. Okay, so these are the type of analysis that we can perform from this set of genes, and definitely we are trying to find out the hub gene. So it is a hub gene. Then we try to understand and analyze the network of this particular hub gene. And this approach where we are trying to analyze the network is known as network science approach to biology. And system biology is a part of that only. Fine. Yeah. So and here like you can see that these are the two genes only which are sharing a network. These are present in isolation. So it means that these genes are not involved like having any interaction with a given set of genes. But these genes are definitely very important genes because these have been obtained as upregulated. So this will have more power of differentiating as compared to these, because when you will be studying machine learning things, you will study a particular concept of known as heteroscedasticity, okay, or multicollinearity. So multicollinearity is a concept in which two different set of features are correlated with each other. And in machine learning or the statistics, we are never expecting or will never want to have two different set of features which are highly correlated with each other when we will be trying to make a classifier or based models. Okay, because सोच देखो यार, अगर मेरा A अकेला क्लासिफाई करने के लिए रिस्पॉन्सिबल है, तो why should I have some variable which is correlated with A? So we'll try to include only one of the features out of the two. This is complete machine learning aspect of things. Yeah. And as I'm telling you this thing here, that after getting this set of networks, we try to go for the enrichment analysis. So enrichment analysis is nothing but simply we are trying to find out which are the different set of pathways in which my given set of genes are enriched in. So this is the level at which a non-biologist will understand. Just try to imagine you are a non-biologist, you went to a doctor and the doctor says that your complement and coagulation cascade is being upregulated. तो आप गाली दोगे डॉक्टर को because you will not understand anything. So okay, so fine. My mistake. Okay, fine. So if the doctor will tell you that your these set of genes are being upregulated, so you will not be able to understand anything. But if the doctor talks in terms of this particular thing, then also you will not be able to understand this particular concept because this is a pure contextual based word. But if the doctor will try to make you understand the meaning of this particular set of pathway, then you will definitely appreciate and able to understand that yes, मेरा complement system pathway जो है upregulate हो रहा है, it means that some type of infection I am having right now. Okay. Similarly, if your P53 signaling pathway is being upregulated, then you will say that might be some those particular driver genes have been changed, which can lead to cancer in my case. So these are the different set of analysis which we are able to understand and interpret only at the pathway level, not at the gene level, because you will not be able to understand the genes. Okay. And definitely we have a large different set of databases. So like we have gene ontology, we have KEGG pathways, KEGG database. KEGG is basically Kyoto Encyclopedia of genes and genomes. Then we have Reactome, we have Wiki pathway, and we have a lot other pathway like databases. So this is at the level of pathway analysis. Yeah. So this is one of the different concept that we can make use of the sequencing data that we get from the labs. Okay, that is, you can find out the set of genes which are differentially expressed. Based upon this set of genes, you can visualize your samples, that how the samples are present, they are present in clusters or not. After that, you can look at the level of protein-protein interaction network and a level, that how these set of genes are interacting with each other. You can look at the enrichment level, that what are the different set of pathways in which my set of genes are being responsible. And after that, you can definitely make machine learning based classifiers which will tell you, help you to fetch only those respected set of proteins which are of very and very high importance in the out of even all the set of differentially expressed genes. And that particular concept is known as feature selection in machine learning. So we can perform all those things, and right now I am writing a paper in which I have made use of all these concepts. Let me tell you. Yeah. So what are the different requirements? Because here I am finishing my topic, because it was just to give you a brief idea how the data is being generated in the labs and how this data is being analyzed at the computational level, and what are the different set of requirements to understand these concepts. So first of all, you should have a very high understanding of data, because you will not be able to do anything if you are not able to understand your data. So you should have understanding of data. You should have a very fair and a good understanding of statistics, not at +2 level only, definitely. So you should have understanding of the graduation level statistics. You should have a better idea of mathematics, both at +2 level and few concepts of graduation level, like linear algebra. Like linear algebra is the heart of machine learning, data science, and artificial intelligence, everything. Because I am a person of, I am a data scientist, so I have to know these things. So, and I'm a mathematician as well. So these are the things that you need to understand first before diving into the mathematics. And then you should definitely have a very good level understanding of machine learning concepts. Like you should know what is EDA. EDA is exploratory data analysis, how to make use of EDA and how to understand your data. Because EDA is the thing with which we start with analysis. So first of all, we perform the EDA upon our data and we try to understand the data. You should have understanding of the dimensionality reduction. So dimensionality reduction was here because we had, suppose that after performing DEG, we got total 1000 genes. But even we have 1000 features or the columns, you can't visualize the data upon 1000 features, because we have the limitation or constraint of plotting the data over only 2 dimensions, the x and the y axis, or maximum 3, that is x, y, z axis, but not at the 1000 levels. So there we make use of the concept of dimensionality reduction and reduce the dimensionality from 1000 to 2, and then we plot the samples based upon these 2 features which are representing those 1000 features. How? This is a ML concept. So you should have understanding of dimensionality reduction. You should have very good understanding of clustering, linear regression, logistic regression, random forest, and SVM. These are the set of machine learning algorithms that you should be able to understand, because these are very highly used in bioinformatics. Trust me on this, these are very highly used algorithms in machine learning. You should not Even if you don't understand deep learning and other aspects, it is absolutely fine. But you should be able to understand clustering at least. You should be able to understand dimensionality reduction. बिकॉज अगर आपको डेटा दे दिया, आपने डायरेक्टली कोई किसी का टूल उठाया, सपोज दैट लाइक लिमा इज अ टूल और डी सी इज अ टूल, आपने टूल उठाया, लाइब्रेरी उठाया, आपने डेटा लिया, आंसर आ गया, it is just useless. None of the industry is going to entertain you. Because Divijay sir is sitting here, he is from industry, he can tell you industry never wants those people who are expert in making use of libraries. But you should definitely able to make use of library, but you should have a very fair understanding of the concepts as well. अगर आपको सिर्फ लाइब्रेरी रन करने आता है, then you will be of no use to the industry. Because अगर आप कहीं पे अटक जाओगे, how are you going to debug that? Okay, because all the different set of libraries are being designed according to the requirements of a particular problem by a particular scientist. Okay, and they are not very highly optimized. Like a lot of libraries are definitely highly optimized, but हो सकता है कि आपकी नीड के अकॉर्डिंग, डिजायर के अकॉर्डिंग कुछ चेंजेस करने पड़े, and that you will be able to do only when you have understanding of these concepts. So so it is one of
Alok,
the,
just to add one thing
yeah, sure, sure.
there. So the thing is, you have a data with you, but you can mine it in a different way to ask a different question.
Definitely.
It's like the data contains everything, but it depends upon our need in what way we are going to use that. You run the different algorithm on the same set of data, and you will have a different outcome out of that. Now it depends upon your knowledge, which one is correct and how far is correct.
डेफिनेटली, बिकॉज डेटा तो अवेलेबल है, बहुत डेटा अवेलेबल है, एंड ट्रस्ट मी ऑन दिस, any of the data is publicly available only when a particular scientist or the research lab will analyze that data. Okay, so you need to design a new question in your mind first, and so that's why we always say that you should, being a scientist or being into the research area, you should always try to focus on why first, and then you should think what, how. Because how तो आप कर लोगे, a lot of people will be there who can help you out how to process these things, but question आपको खुद ढूंढना पड़ेगा. Am I right, sir?
That's the basic reason I talked about the DNA RNA in the like second slide itself.
Right.
We know about it, but how it's changing our like future clinical services, that came from that one too. Uh, Everyone can do the sequencing and other stuff, but even there are like instances where your sequencing get failed, and if you don't know why, then you cannot fix that. You should know like where things may have went wrong. So these are the things. We are knowing about DNA RNA for like 1953 or 54, but the real use is like coming in last 20 years when people thought out, okay, these are the basic structural things. If it gets something wrong inside it, we are starting having problem. So we should know that.
Definitely. So I always say, and like everyone in the research field says, so start asking why before starting any question. Okay, because otherwise, what you will do, like I myself do a lot of times, बतेरो एनालिसिस कर लेते हैं, एंड में समझ ही नहीं आता क्वेश्चन क्या पूछ रहे थे हम. सो ऑलवेज ट्राई टू फ्रेम अ क्वेश्चन फर्स्ट एंड देन ट्राई टू लुक एट द कॉन्सेप्ट. वो कॉन्सेप्ट लिमिटेड है, ठीक है, कॉन्सेप्ट तो लिमिटेड है, बट आपको क्वेश्चन अनलिमिटेड होंगे जो आपको सोचने पड़ेंगे एंड ढूंढने पड़ेंगे. सो स्टार्ट लुकिंग एट द क्वेश्चन. सो येस, एट द एंड आई वुड लाइक टू, ये क्या हुआ, बट इज नॉट मूविंग, या फाइन, या. सो दिस इज वन ऑफ द बेस्ट स्लाइड फॉर anyone who always gives a presentation. That is, thanks for listening patiently to us and being at the end till the end of the session. And so I'd like to have any questions, anything, any suggestions, and definitely your feedbacks that you would like to have, and definitely please and please feel free to ask your questions now. Okay, so I'm done from my side as well. So yeah, so now we are most welcome to your questions that you have in your mind. So I'm done from my side as well. Thanks. So hi Tanav, are you there? Hello Tanav, are you there?
Hello. Yeah, yeah, I'm here. I'm here. I
Yeah,
was
hi
just listening
Tanab.
to you.
So Tanab, tell us like, do you have any question in your mind? Because you are from a non-biology background and you joined in the at the right time of the session. Because when I was
Yeah,
in
yeah,
the analysis part of the session, which was pure machine
yeah.
learning based concepts. So do you have any question in your mind right now? Please feel free to ask.
No, no, not really, because these concepts, I think we've discussed them before as well while we were working on our project, and the similar concepts were going on. So you've cleared most of my doubts beforehand. So
Right.
that was pretty cool.
That's great. So, because you are from a non-biology background, so can you give an idea like how can you make use of your computer science background knowledge into the field of biology?
So I feel as a data scientist or a computer science student, data is everywhere. It could be anything: biological data, agricultural data, data of websites, anything. So if you know certain mathematics and machine learning algorithms, you can implement that just by studying the data and performing a little bit of EDA. Even if you don't know what the data exactly represents, those graphs and figures do help out. Obviously, there are biologists like you who are there to guide us and help us around wherever we get stuck.
Okay, that's nice. Fine. So, great, yeah. So then, guys, please feel free to ask your questions, yeah. Like otherwise, it will be just a, not that fruitful to us. Okay. So let me ask.
Alok,
Yes, sir.
just to interrupt, I have some other thing to look into it, like some lab work. So if
Sure,
I
sure,
am
not
not
a, not
needed
sure,
here,
sure, because we are done from our
uh,
side, so you can drop off the call. Sure.
thank you guys for listening me, and hopefully it will help you in some way.
I will share the question if they have any to you over to you. Okay, sir. Over on
Yeah,
that.
yeah, you can make that sheet and
Sure, definitely,
send
definitely.
them.
I will. Sure, sure.
Okay.
Thanks a lot
Okay
for your
then.
time.
Bye everyone. Yeah, bye.
Thanks, sir. Yes, to the you have raised your hand. Do you have anything to say?
Yes, sir, but I have to ask one question from Narayan, sir, but I think he has left.
हाँ बोलो क्वेश्चन बोलो एक बार like I will try to send the question to him.
Yes, sir. Sir, my question was like, sir explained that we are using RNA sequencing for like cancer gene analysis in our clinical trials.
Hmm.
So, my question is like, how we will interpret that we have to do whole genome sequencing or the exome sequencing?
Okay, so this question I can answer definitely. So exome sequencing we always go for when we have a very when we are trying to ask the question from the coding region only. Okay, but when you are not that sure and you are open to questions, when you are open to questions and you are open to as many analysis that you can think of, when you do not have an exact question in your mind. Okay, so and you and your answer can lie in the LNC RNA, that long non-coding RNA, or into the non-coding regions, into the introns, or into another aspects. Then you always go for the whole exome sequencing. As whole exome sequencing is a very costly thing, we should always try to protect ourselves not going to whole exome sequencing. Only go there when you have a very like when you're not able to figure out when you are interested in non-coding regions first of all. Okay, and when you are interested in a more open question, then you should go there. Got it, Stuti?
Yes, sir.
चलो great यार. So guys, it will be like fine. So let me एक मैं ही पूछ लेता हूँ यार, ऐसा तो आप बोलोगे नहीं. So then we have Ekta here. Hello, hi Ekta, are you there?
Yes, yes, I'm here.
Hi Ekta. So can you give your feedback on the session?
Yes, it's interesting. Earlier, I thought that as a bio student, it's tough to learn these computer things. But now I'm getting interested in these things.
So that's nice. So hello Vivek, are you there? Hello Vivek, are you there? Might be he is not there. Hello Manvi, are you there? Hey, hi Manvi, are you there? Hello. Manvi, you're not audible actually. Might be you're on mute or something.
Am I audible
Yeah,
now?
now you're audible. Yeah. So can you
Okay.
share your
So
feedback or any question that you have?
yeah, the session was pretty good and basic. So I would give you one to feedback that
if in future, I mean, I would be looking forward to other sessions also. But if the sequence data analysis, that the part you covered in a short bit of span, that you can maximum information that you covered,
Okay,
that
so
was quite great. But it would have, I mean, I would want to explore more on that part.
on the
um,
analysis part or on the basic sequencing part?
the analysis part, it was quite good, the basic information that
Okay,
you
okay,
gave
okay. All
today.
the analysis that is after, like
But I
differential
think,
expression, PPI network,
yeah,
ML
yeah,
part, and all, right?
but yeah, I think due to the time constraint, you have just made it a little short. So I would want it to be more explanatory. I
Definitely. So
mean,
let me give an idea. So, hello sir, I'm traveling. Cannot mute. It was an insightful session. I'd like to attend. Yeah, fine. So thanks, Vivek. So like we have your email ID. So we keep on conducting these type of sessions. So let me tell you. So our next session is 22nd only. So next week only, and in that session is a very generalized session. So the topic of that session is like why to go for a PhD. Okay, and why should I go abroad for a PhD? Why shouldn't I pursue a PhD from India? Or any general question that you have in your minds. And there will be two or three speakers for that session. That will be a more panel-like type of discussion. That will be just question answering sessions. And the speaker for this session is from Italy. So she has completed her PhD from Italy only. Sorry, yeah, Italy only. And currently she is placed at US for a postdoc. Okay, so she will be there along with a few other persons. So we'll be trying to tell you why to do a PhD. Okay, and क्यों ऐसी बनी हुई है एक theory that PhD मतलब life बर्बाद. ठीक है, so we'll try to bring such a session which will help you out why to pursue a PhD and what the different application type of things that are to be followed while applying for a PhD. Okay, and after that on over 30th we are trying to conduct another event where we will be having the topic of the talk will be the history of AI in healthcare and how to become a medical data scientist. So that is basically more computational thing because that will be complete ML AI part. And the speaker for the talk is from Switzerland. So he's completed his like BTech from IIT Kharagpur and he did his PhD from Switzerland, Roche University. So he'll be, and he is a medical data scientist only. So he'll be shedding light upon how to be a medical data scientist, which was a very insightful talk. Because I am also in the process of that only, but I always love to have people which are expert in their domains. So we're trying to build a community where you will be able to interact with those type of people. And definitely, like due to the time zone differences, we were not able to conduct this session in the daytime because in the daytime if we would have conducted this session definitely much more participants would have been there, but due to the time zone we are also constricted to this. Okay, so if you have any questions feel free to ask me your questions. Okay, and if you don't have any question it will be really great if you guys could please switch on your camera for 1 minute so that I could just pick a pic of this particular team who are sitting here right now, and then we could disconnect the session. Okay, so if any of you have any question feel free to ask me first. Yeah, Himangni, you are there. Yeah, Himangni, do you have any questions in mind?
no, I do not have any questions. It was quite clear. Thank
Oh,
you.
that's great. Himangni, do you have any question or something? Madhu, she have just joined and went off somewhere. Yeah, fine. Yes, she is there.
Hello.
Yeah, hi Himangni, you're audible.
Actually, I am so late, so I don't think I have a question because I don't know the topic actually.
Okay, so you joined at
Uh,
the end. Okay.
I have missed the entire thing, so
Oh, that's not a big deal. Like I will be uploading this session over the YouTube channel. Most probably within 2 or 3 days. Okay.
okay, thank
That's so
you.
great. Yeah. So guys, it's humble because if you feel comfortable to switch on your camera, please switch on your camera so that I could just take a pic, and after that we could disconnect the session. Hey, hi Tanav. क्या हाल है बॉस?
बढ़िया बढ़िया आप बताओ.
ग्रेट यार, हम तो बढ़िया रहते हैं. या एक तो कैन यू प्लीज स्विच ऑन कैमरा? Oh, that's great. Hi. Manvi Stuti and Magni, you also good please switch on. It will be great यार. Manvi, are you there? क्या कर लो प्लीज.
I think there's a network problem. I couldn't do that.
Okay,
I'm
not an issue.
trying, but it's not
Okay,
working.
not an issue.
Sorry.
Stuti, are you there?
Yes, sir, but there is no electricity
Oh,
actually.
that's nice. That is awesome. ठीक है, so not a big
Sorry,
deal.
sir.
ठीक है, so let me capture a pic because my phone is switched off, so I'm capturing it from a tab. So
टैब साइड
Tanab,
कर लो आप ही
Tanab,
नहीं.
can you like मेरी शक्ल तो गायब हो गई, so Tanab, can you also
वही
take a
वही.
pic and then share with me?
हाँ, I'll do that.
Yeah, thanks, sir.
Daily.
Clearly, oh, that's great. Yeah, ठीक है. So thanks all of you for joining, and looking forward to in connection with connected with you. And feel free to reach out to me any moment of time whenever you need any help or something. Because I'm a very friendly person. Tanav knows very well. Okay, so chalo bye, take care. Yeah. So if you have anything you mind, you can stay in the call, and others can disconnect off the call. Yeah. So thanks. So bye,
Thank
take
you.
care.
Bye.
Bye. Yeah. Manu, do you want to say something?
No, no, just saying bye.
Oh, chalo
Thank you.
bye, take care.